So good evening, everybody, and welcome to today's episode of Marvelous Medicine. It will be focusing mainly on gut microbiome. The speaker is Dr. Sree Subramanian. He's the consultant gastroenterologist at Cambridge University Hospital, NHS Trust, and is also the honorary senior lecturer at the University of Liverpool. So Sridhar did his MBBS from Jipma Pondicherry and then subsequently trained in Lincolnshire, New Zealand, and Liverpool. He has done a lot of research on inflammatory bowel disease, and is also part of research which is funded for colitis and Crohn's disease. This is microbiome is a pretty new topic for all of us, and we really hope to gain some insight from you, Sridhar.
Okay.
Please feel free to add to your introduction while you speak. We are very much honored to have Dr. B.S. Ramakrishna to moderate this session today. He's a director and senior consultant at the Institute of Gastroenterology, Hepatobiliary Science and Transplantation at SIMS Vadaparani. His research interests are inflammatory bowel disease and microbiome. He's published 170 articles, 20 book chapters, and also holds a patent. He's the editor-in-chief of Gastroenterology, Hepatology, and Endoscopy Practice, and he's been the past president of the Indian Society of Gastroenterology. Thank you, Dr. Ramakrishna, for agreeing to join us.
Thank you.
Joining him will be Balamurugan Ramadas. He's the additional professor, biochemistry, and associate dean of research, and head of Center of Excellence for Clinical Microbiome Research, and in charge of the Clinical Biochemistry Laboratory at AIIMS Bhubaneswar. Balamurugan did his BSc Lab Technology and MSc Biochemistry from Jipma Pondicherry, and his PhD in Medical Biochemistry at CMC Vellore. As you would have understood from the introduction, he's very much involved in research on microbiome. Really nice to have you, Balamurugan, and hope we have a very good discussion at the end of Dr. Sridhar's talk. Over to you, Sridhar.
Okay, yeah, great. Let me just try and... Is it working, the screen share?
Yes, yes.
Yeah, great. Thank you very much. Actually, it's a huge pleasure to be doing this. And particular thanks to Vidya, actually, because she's been doing a stellar job of running this series, and it's not easy to get this organized, I would imagine. And particular thanks also to the moderators for giving up their time, and to all the audience. So I want to start with a couple of disclaimers, really. The first one, and probably the most crucial one, is that I'm not a card-carrying gut microbiologist. And I'm glad that actually Bala is here to maybe field some of the strong technical questions that may arise.
Sorry to interrupt you, Sri. Can you get closer to the mic?
Oh, yeah, sure. Okay, great. Is that a bit better?
Yes.
Oh, great. I was actually hoping to steal my son's AirPods, but I couldn't get it to work. So I'm stuck with the old-fashioned thing of speaking into the computer. So I just wanted to start with a couple of disclaimers. Firstly, that I'm not a card-carrying gut microbiologist, so I don't do gut microbiome work on a day-to-day basis. But I've done a lot of research in the past, mostly to do with single organism gut bacteria, but I'm glad that our moderators are here on hand to help with any strong technical questions that may arise at the end of this. Rather unashamedly, the discussion is predominantly going to be GI focused for obvious reasons. Firstly, because it's to do with gut microbiome, and secondly, to do with the fact that I'm a gastroenterologist. So if we look at the popular news these days, there's a lot of news on gut microbiome in general. So these are a couple of representative screenshots from Times of India, one focusing on gut bacteria and heart disease risk, and the other one more recently on COVID, not just on COVID recovery, but also on other aspects of COVID. So there's a lot of news in the public media about gut microbiome, and I would say that the public interest in gut microbiome and how to modulate that is increasing constantly. But actually, if you look back in the prism of history, this is not entirely new. And Hippocrates, allegedly some 2,500 years ago, had stated that all diseases begin in the gut. And I think that is a fairly visionary hypothesis. And if you look at the number of diseases that have been incriminated to gut bacteria, and I think he was probably quite right to state that. So I think it's timely to sort of look at gut microbiome, and hopefully I can provide a window into gut microbiome and what it means in the pathogenesis of all these diseases, and potentially how we can think about changing the gut bacteria. So I'm going to start with a simple introduction to the gut microbiome. I'm fairly conscious that the audience today are a wide spread of different background of clinicians. So I just want to start with the basic glossary of the key terms that we'll be encountering over the next half an hour, 45 minutes. So these two terms, the top two terms, microbiome and microbiota, have been fairly used loosely interchangeably. So just to clarify, microbiota refers to the collection of microorganisms present in a defined microenvironment. Predominantly, when we talk about microbiota, I think subconsciously most people refer to bacteria, but although I think a key part of the microbiota is things like fungi and viruses, and a lot of evidence is emerging for the role of fungi and viruses, for example, in inflammatory bowel diseases. But in relative terms, the literature on fungi and viruses are still few and far between as compared to the role of bacteria, which is way ahead. Microbiome, on the other hand, it includes the microorganisms and their gene products and their genomes. So the terms are used interchangeably, but there are some key distinguishing factors between the two. Operational taxonomic unit, we won't use much of that, but essentially the best way to think of that is a family of relatives, so to speak. So these are similar microbes that are grouped into a single unit. Metabolome, again, this is an emerging concept over the past 10-15 years. That is the product of metabolites that are derived from microbial metabolism, predominantly of carbohydrates, proteins, and lipids. Then I want to introduce two key concepts which are going to be central to understanding today's talk, which is diversity and richness. We are used to the term diversity. When we refer to diversity, we talk about human diversity. That would mean, say, for example, people of different ethnicities, different colors, different backgrounds, living in a particular city. And microbial diversity is very similar. So it refers to, for example, how many species are there in a niche. And not just that, it also refers to the distribution of the species. So, for example, in the pictorial cartoon on the right-hand side, you can see that A, B, and C, the species of plants are distributed evenly. So good diversity means that there is a lot of richness, i.e. a lot of different bacterial species, but it also means that the species are distributed fairly evenly. A dysbiotic state would be that the species is dominated by one particular organism or species, or a few different organism or species. So there'll be a lot more of A compared to B and C. So when we refer to a diverse group of microbiota, we refer to not only the richness, but also the community structure where there is a lot of even number of bacteria, for example. So these are key terms to understanding gut microbiome in the context of health as well as disease. So just to set the context of gut microbiome, it's probably easier if we think of the gut microbiome as a virtual organ. Just in the way we think of liver and spleen, we think of the gut microbiome as an organ, except that it's not a visible organ that's described in textbooks. Instead, it's an invisible organ, but it's useful to think of it contextually as an organ because actually it has a lot of microbes, around 100 trillion microbes in the human intestine alone. The number of gene products is quite staggering, really. The humans, in general, we have 22,000 genes, whereas the microbes have 3 million genes, which is quite a lot as compared to the human genes. They apparently weigh around a couple of kilograms, and there are at least 1,000 different species of bacteria, although with modern sequencing technology, that number keeps going up and up. So there's been a lot of focus over the years about the early phase of development and how critical it is. So, for example, whether or not the mode of delivery makes a difference. So when the infant is born, within first 20 minutes, the colonization of the gut bacteria happens. And there are some fundamental differences between babies who are born by cesarean section versus vaginal delivery. But that difference seems to even out after 9 months. Infamously, there is a South American obstetrician who was administering vaginal juice to newborn infants in the hope of colonizing the gut with a vaginal microbiome, which is generally perceived to be healthier. So after 20 minutes, you acquire the microbiome either from the vagina or from the mother's skin, which tends to be the characteristic microbiome in cesarean section infants. And once again, there is a difference between breastfed and bottle-fed infants. They have different sort of bacterial species. And then when the baby starts weaning, then you get more of a transition towards more of an adult state microbiota. And after that, it pretty much tends to stay stable for many years, except when you become elderly, when again, the diversity of bacteria reduces, diversity of the microbiome reduces when you become older. So in the gut, the distribution of gut microbiota follows oral to anal gradient, meaning that the amount of bacteria really increases to the order of trillions once you get to the colon. So the colon tends to have the highest density of microbiota as compared to the ileum and jejunum, whereas the stomach is relatively free of microbiota due to stomach acid. The colon is heavily populated, and a gram of stool, for example, can have as much as 1 trillion bacteria. In general, in healthy humans, the bacteria are ordered into different phyla. So the major phyla are Firmicutes, Bacteroides, Actinobacteria, and Proteobacteria. But as you can see from this table, the predominant, i.e. 90% of the gut bacteria or 95% of the gut bacteria belong to either Firmicutes or Bacteroides, and a smaller proportion belong to Actinobacteria and Proteobacteria, for example. Of course, in dysbiotic states, like, for example, in inflammatory bowel disease, this could be reversed and there could be a dominance of Proteobacteria compared to Bacteroides or Firmicutes. But in a healthy condition, this is the distribution of the bacterial phyla. And of course, the gut bacteria are not just there hanging out. It is a truly symbiotic relationship. The job of the human host is to provide nutrients in the form of ingested carbohydrates and fibers. And then the job of the microbiome in turn is to actually do a number of key functions. Firstly, it harvests energy from the food sources. So, for example, fibers can be fermented in order to produce short-chain fatty acids, which are quite critical in nourishing the colonocytes. They also synthesize some vitamins, and more crucially, they actually provide a key balance between the gut immune system, and they constantly are in dialogue with the gut immune system such that you get a healthy state of tolerance. So the gut microbiota really have a number of key functions. And the functions are not just limited to the gut. And in fact, there is a thing called the gut-brain axis, where there is a constant feedback between the gut and the brain, where there is a bidirectional relationship between the gut microbiota and the brain. And this may in part explain why diverse conditions such as, for example, Parkinson's disease and dementia are associated with disturbances in gut bacteria. So there are a number of different key functions that the gut bacteria have in a healthy situation. So next we come on to what really constitutes a healthy microbiome. And this is quite a tricky and complicated area because it's critical for us to understand what constitutes healthy microbiota when we come to interpret it in the context of disease states. And it's incredibly complicated, partly for the simple reason that there are a number of different factors, even in healthy people, which can influence the gut microbiota. We have already spoken about the mode of delivery, but there are also a number of other factors, such as, for example, geographical location, and we'll come on to that in a minute. Host genetics, so if you get identical twins, they do share some gut bacteria, although environmental factors are more important in that context. So the state of the intestinal, speed of the intestinal transit also influences the gut microbiota. So if you can imagine, there are a number of different factors that govern what a healthy gut microbiome looks like. And that becomes really, really difficult in the context of how this can vary from one person to the other to define what's a healthy human microbiota. But thankfully, technology has improved as well, which has meant that we can actually study these things in greater detail. When I was a student, for example, we were used to doing blood cultures on agar plate. And in fact, when I was doing my research around 20 years ago, 20 years ago, we're still doing this old-fashioned method of bacterial culture, growing things on agar plates, and so on and so forth. But that's largely been superseded by these high throughput technologies. So you can sequence a whole bacterial population by using 16S sequencing or shotgun sequencing, et cetera, and you can actually get a comprehensive catalog of the bacterial population within either a stool sample or a salivary sample or whichever sample that you have. So these high throughput technologies have really helped us to characterize the bacterial population in a lot more detail. High throughput, not just in terms of gene sequencing, but also in terms of metabolic characterization. High throughput metabolomics or metabolomics means that we can actually study the metabolite that these gut bacteria produce in a particular sample. So it's really opened our eyes to how complex the gut bacteria are. And therefore, exploiting this technology, a number of research groups across the world have tried to build a catalog of what the human gut microbial gene sequence looks like. So the first effort, first significant effort was probably done by this Chinese group who published their paper in Nature some years ago now, 12 years ago. And it was a mammoth effort of characterizing studying 3.3 million genes from the feces of 124 individuals. They took healthy people as well as people with inflammatory bowel disease and looked to characterize the entire bacterial genome. And when you do that, you encounter this incredibly complicated network of bacteria within the stool. So as I explained previously, predominantly it's dominated by Firmicutes and Bacteroides, and you can see the yellow circles are Firmicutes, the pink ones are Bacteroides, and that constitutes roughly around 90% of the stool microbiome. A smaller proportion are the Proteobacter and the actinobacteria, but this is how incredibly complicated it looks in a healthy situation. And the next significant effort really was the Human Microbiome Project, which is done by Jeffrey Gordon and colleagues using funding from the National Institute of Health. And here they did a massive exercise, really looking at the gene sequence of bacteria within the stool, within saliva, and a range of other biologic samples. And once again, they found similar sort of things, that most of human gut bacteria belong to 4 phyla, which I've already explained. But there are some key inter-individual variation, and that variation is largely driven by Firmicutes and Bacteroides, i.e. the 2 most dominant kind of phyla. So there are variations between individuals, which makes it harder really to fully understand what a healthy microbiome looks like. Contrast that to the genome, the human genome, where there is very little variation. So there is 99% similarity in the genome among individuals and less than 1% variation. Whereas when it comes to the microbiota, there is around 80% variation and 20% similarity. So that makes it very, very complicated to define a healthy microbiome. But equally, there are some advantages because you can leverage it to develop some personalized medicine. Where there is a lot of variation, you can develop precision medicine much more robustly. So just to answer the final point as to what constitutes a healthy microbiome, we still don't know. In fact, there was an international consensus group that got together a few years ago to try and nail this point down as to what constitutes a healthy microbiome. And in the end, actually, they came up with a few core points, which is that the markers of human health and microbiome structure in human health is not fully established, which is what we discussed. But they've come up with a lot of recommendations as to how we can study this further. And importantly, they said microbial communities are highly individualized, saying that there's a lot of variation between individuals, and how not only that, how an individual microbiota responds to, say, for example, a course of antibiotics might be fundamentally different from one person to the next. And that explains why some people get antibiotic-induced diarrhea and some others don't. But a basic simple rule that has been shown repeatedly in different studies when they compare healthy and diseased individuals is that if you have a diverse group of bacteria, it is better. Just like human diversity, which is good for communities, microbial diversity is good for human health. So the more bacterial species that you have and the more evenly distributed they are, the better it is. And that's pretty much what we can say about what we think of as a human healthy microbiome. In the next few minutes, I want to focus on how the gut bacteria impacts on diseases of modernization. As you know, there is a tremendous difference in disease patterns between industrialized countries and non-industrialized countries. And this is really a striking graph of the incidence of different diseases over time. So, for example, some of the communicable diseases like tuberculosis and mumps have gone down dramatically in the past 50 years in industrialized countries. But if you look on the right-hand graph, we have just traded simply one set of problems with another. So we have got rid of all the infectious problems, but then we've got non-communicable diseases, which are on the rise in industrialized as well as newly industrialized nations. And this may, in some part, actually be linked to gut microbiota, because it's not as if the genetics of people have changed dramatically over the past 50 years. So there's got to be some environmental factor which is driving up these diseases and the incidence of these diseases. And one place that's quite different in terms of humans is the gut microbiota. So we can see that actually, if you live in distinct geographical locations, the gut bacteria tend to be different. There is no universal consensus about this. And in fact, a German group previously said that no matter where you live, you belong to one of 3 distinct enterotypes, which are dominated by either one of 3 different phyla. But notwithstanding that, several other studies have shown actually that there is a distinct difference between microbiota in Western countries as compared to newly or non-industrialized countries. And this may in part link to the diseases that we are seeing. And here's one example of that very interesting study, which was published in Cell Report some years ago now, which systematically collected stool samples from Papua New Guinea, 2 different tribes in Papua New Guinea, which is a which officially apparently is the remotest part in the world, and then compared that to actually a non-industrialized place in the US, and then showed that there are differences in terms of Western microbiome versus non-industrialized microbiome. And the other thing to note here, key thing to note, is that you can see on the left-hand side of this dot plot that in Papua New Guinea, all these dots are fairly close to each other, whereas in US, all these dots are fairly spread apart. What essentially this implies is that in a given population in a non-industrialized country like Papua New Guinea, the microbiota of different individuals are closely related to each other. This might in part be related to cultural aspects, where people in remote environments tend to get together quite a lot more. They tend to hang out in the same places, etc., which means that there's quite a lot more room for bacterial dispersal and bacterial sharing. Whereas Western community in general, probably a bit more dysfunctional, and people are just in their own rooms, watching TV at night, etc., locked away from each other, which means that there is not enough room for bacterial dispersal, and that the bacterial populations are quite different. So not only is bacterial population different between East and West, but it's also different between individuals within the West itself, because of the lack of mixing and lack of bacterial dispersal. A similar sort of finding was found in the mammoth exercise again, which is a study where actually people from the scientific community contributed fecal samples, predominantly from the US and the UK, but also from Australia, as well as other countries such as India, for example, and Vietnam, and so on and so forth. And here again, they found some fundamental differences between industrialized countries versus people who live in non-industrialized countries, where you can see, based on this bar graph, again, there is fundamental differences between the two different populations, where there are remote farming community and hunter-gatherer communities have a different set of bacteria as compared to industrialized countries. So I won't labor the point too much, but essentially it looks as though there are some differences between westernized versus non-westernized countries in terms of bacterial populations. And it gets even more interesting because in this study, which is published in Cell some time ago, they systematically looked at people who migrated to the US from different countries. I think in this case, it was China and Vietnam, where there is people from a remote tribe, both from China and Vietnam, who migrated to the States. And of course, people who migrate to the States from these communities tend to have a higher incidence and prevalence of obesity. So I think underneath it all, I think the authors wanted to investigate the hypothesis that gut microbiome in some way is related to migration, and in some way is also related to the body mass index. And interestingly, I think they found some fairly intuitive kind of results, where they found that soon after you migrate and the longer you've been in the States, then you start to fundamentally change the bacterial phyla. So for example, Prevotella, which is a predominant bacterial strain, which has a lot of fiber degrading properties, that tends to gradually diminish over time, and then it's replaced by Bacteroides, which is more predominant in Western population. So you're tending to trade off one set of bacteria to another, and that may just be a reflection of diet, where your diet has changed as you move to a Western world. And then that brings us to the question as to why there are these bacterial differences between the East and the West, between industrialized and non-industrialized countries, and whether or not some of this could be explained by some of the diet and lifestyle factors. And of course, diet is a key driver of gut bacteria, and that's been shown in different studies, as well as other things like antibiotics and different medications, for example, which can influence the bacterial population. Equally, specific components within the diet, for example, emulsifiers such as carrageenans and polysorbates, which are quite widely used in diet to prolong the shelf life. So for example, a lot of processed foods use a lot of emulsifiers. And in this very elegant study, which is published in Nature some years ago, they used mice model and then showed that if you add emulsifiers to the diet of mice, the mucus layer in the colon becomes very thin. That means that more bacteria penetrate the mucus layer into the intestine and then probably cause inflammation. And not only that, when you add emulsifiers, there is an increased incidence of obesity and metabolic syndrome in the mice, as well as increased incidence of increased severity of colitis. So there are factors in the diet, specific factors in the Western diet, such as addition of emulsifiers, for example, which may drive the bacterial differences that are observed in these two different populations. So now that we think we understand what gut bacteria and health looks like, we'll move on to gut microbiota alterations in disease states. So this is just a graphic of what happens in health and disease. So in the healthy sort of situation, there is a higher intake of dietary fibers, for example, which then drives the production of short-chain fatty acids and antioxidants. And I've already explained that these are critical in nourishing the colonocytes and decreasing inflammation. Whereas if you, in a disease situation, exactly the opposite happens, where you have a lot of animal fat intake, which drives the production of adverse compounds like free radicals and trimethylamine, for example, which increases the risk of atherosclerosis and so on. So you get fundamentally differences in terms of gut bacteria as well as metabolome in these diseased and healthy states. Like I alluded to right at the beginning, there is a number of disease conditions where there are associations between gut bacteria and diseases. I won't go through each one of them in the interest of time, but suffice it to say that actually in organ systems as diverse as the brain and the intestine, and they couldn't be further apart from each other, you get diseases which are associated with disturbances in gut bacteria. One of the earlier studies which caused a lot of excitement in this field is whether or not gut bacteria could actually drive obesity. And in the study, which is published some years ago now in PNAS, they looked at the mice from obese mice as well as lean mice, and then found that there are differences in terms of bacterial population. In obese mice, for example, there is a 50% reduction in Bacteroides, and in lean mice, there was a reduction in Firmicutes. In fact, some authors, including I think Bala, has proposed using this as a biomarker of obesity, the ratio between Firmicutes and Bacteroides. And obviously this begs the question, as to whether or not you can actually treat obesity by just altering this kind of bacteria. And in fact, that's exactly what people have done. So people have done a number of elegant experiments, predominantly in mice, where they actually take the microbiota from lean mice and inject into obese mice and obese mice into lean mice, vice versa, and then showed that the phenotype of obesity is transferable, at least in mice, and it's dependent on the gut bacteria. But similar efforts to reproduce that in humans has not been very successful. And I think we can all agree that mice and humans are different. But when they did the sort of bacterial transplantation from lean people into obese people, although there were some biochemical indices which improved, for example, insulin resistance, unfortunately, the obese people didn't become lean just with a simple intervention. So it appears as though things could be far more complex. And we might obviously be missing the elephant in the room, which is that the calorie intake is another key aspect of weight control. Moving on to more examples of diseases, and I'm going to focus the next few minutes on inflammatory bowel diseases, where this is although I'm focusing on IBD, this kind of picture has been shown in other diseases as well. It's where we spoke right early on in the talk about diversity, meaning that diversity, a diverse set of bacteria is one where you get a lot of different bacterial species. But if you compare a healthy and a Crohn's patient, and here you can see the size of the circle, you can see that instantly the size of the circle is much smaller in Crohn's disease as compared to healthy people, i.e., there is a reduction in diversity in Crohn's disease as compared to healthy people. And I mentioned this a little bit before, where actually there is, for example, an increase in certain types of bacteria in Crohn's disease as compared to healthy people. And there have been some specific examples, and I spent some time looking at E. coli in Crohn's disease many years ago, where there's been a series of several publications which have shown that E. coli, which are non-pathogenic E. coli, i.e. they don't belong to enterotoxigenic or enteropathogenic E. coli, these are harmless E. coli, which have no pathogenicity genes, are highly predominant in Crohn's disease as compared to healthy people. So these E. coli can actually invade the cells and then cause quite a bit of damage. And then they can actually replicate within macrophages, which may lead to some of the suppurative complications that we see in Crohn's disease. But again, though, the ultimate proof of whether or not an organism is causative in an illness is to do a randomized trial trying to get rid of this bacteria. And in fact, we just did that study targeting E. coli within macrophages using a cocktail of antibiotics and hydroxychloroquine, and showed modest benefits, which are comparable to steroids. So I don't think we have found the full answer yet, but it looks as though there might be some organisms which could contribute particularly to the pathogenesis of certain diseases. So it's not microbiome, it's probably microbiota as a whole, and within that there are some species which are worse than others. And the similar story again, unlike E. coli, which is increased, this is Faecalibacterium prosnitziae, which is typically seen as a probiotic bacteria or a friendly bacteria. So if there's a reduced amount of this Faecalibacterium prosnitziae, then it's associated with recurrence of Crohn's disease after people have had an operation. So it looks as though in inflammatory bowel disease, at least, there is reduced diversity, a few candidate organisms which could be driving disease, such as E. coli, such as Faecalibacterium prosnitziae, and so on. Then what's interesting, though, is that actually some of this gut bacterial traits could be transmissible. So in this very nice study, what they did is they collected stool from healthy donors, 15 of them, and collected stool from IBD donors, and then injected them into mice which are predisposed to getting colitis. And the results that we saw are quite interesting. When you actually inject the stool of healthy donors into these mice, they don't get as severe a colitis as when you inject the IBD stool into these mice, and they get a much more severe form of colitis. So implying that actually there are specific bacterial populations within IBD that could be fundamentally important in causing inflammation, and then bacteria are probably central to the pathogenesis. I just want to spend a couple of minutes on gut microbiota and cancer, because this has been very topical. And there is a lot of studies which have incriminated specific bacteria, for example, Fusobacterium in colon cancer. But more recently, there's been a set of stunning papers, really, which suggest that gut bacteria may fundamentally drive the response to chemotherapy as well as immunotherapy. We know that immunotherapy has really taken off in the past sort of 10 years or so, where things like pembrolizumab and ipilimumab, which have been approved in a number of different cancers. But it looks, and obviously with immunotherapy, you get immune-related side effects such as colitis and hepatitis and so on and so forth. But here you can see there are some interesting studies that have been published over the years with immunotherapy. Firstly, that patients who are prescribed immunotherapy, but who also have antibiotics at the same time for a chest infection, or in some cases where cetrin is prescribed as normal, the mortality risk after you adjust for tumor staging is very high in people who have had antibiotics compared to people who haven't had antibiotics. So that immediately raised the question as to whether or not actually antibiotic-induced changes in gut bacteria may be driving this kind of response to immunotherapy. And when you look closely, there have been several studies, and this is an example of some of these studies, which say that actually there are some bacteria which are associated with anti-tumor response. So for example, some of the same things like Firmicutes and Faecalibacterium prosnitzii, which mediate the anti-tumor response, i.e. people who have a good response to these tumors have different set of bacteria as compared to people who don't have a good response to these bacteria. And interestingly, though, if you look at it, the toxicity, on the other hand, is the exact opposite. So, for example, bacteria which are associated with anti-tumor response, Firmicutes, is also associated with the side effect. So, if you get colitis, it may not necessarily be a bad thing, and this has been shown in other studies. If you get colitis with immunotherapy, then you tend to have a better response to immunotherapy as compared to people without colitis, even after adjusting for tumor stage. So again, it underlines the point that actually gut bacteria may be important in driving response to cancer therapy, such as immunotherapy and chemotherapy. Oops. But then the really important and the nub of the matter is proving causation. Okay, so one could argue that, we have seen all these disturbances in bacterial population between disease states and healthy states, but how the hell do you prove that it's a gut bacterial disturbance that caused the disease in the first place? And here's where we get into a classical sort of chicken and egg situation. We don't know what came first. Skeptics of this argument of gut bacteria suggest that actually the dysbiosis is just an epiphenomenon. It probably came after the disease rather than before the disease. It's very difficult. It used to be a lot simpler back in the day. And just as a reminder, this is the Cox postulates, where it stipulates a series of steps that has to be fulfilled for an organism to cause a disease. So this is a famous German physician who came up with these 4 fundamental steps. Firstly, the microorganism must be found in diseased organisms. Then it must be isolated from that and grown in pure cultures. Then it should be introduced to the healthy animal and shown that it can actually cause disease. And then once you treat them with antibiotics, then it should get better. So these are the Cox postulates, and this is the way you can actually prove that a bacteria or an organism can cause disease. And this, of course, not so long ago, Warren and Marshall, who won the Nobel Prize for Helicobacter, this is exactly what they did. They fulfilled the Cox postulates to show that Helicobacter causes peptic ulcer disease, and for their efforts, they won the Nobel Prize. But actually, if you're talking about a single organism, this is very easy to prove in many ways, like in the case of Helicobacter or in the case of cholera, for example. But when you go into a situation where you're talking about thousands of organisms and probably perturbations in community rather than individual organisms, it becomes a lot more complicated. But this is the equivalent of modern day Cox postulates, which actually is not so easy to prove causation. So the first thing we do is to actually take people with disease, for example, let's assume inflammatory bowel disease versus healthy controls. And then similar to genome-wide association studies, you do an untargeted microbiome and the metagenomics, you do all these different sequencing stuff, and then you identify a group of biosynthetic gene clusters. I mean, these are genes and gene products that are intricately linked, and then you identify that these are differentially expressed between people with disease and without disease. And once you do that, then you do some animal experiments where you actually pretty much fulfill the Cox postulates, again, where you take control animals and germ-free animals. So these are mice without any microbiota on any of the body. And then you inject this combination of genes and gene products and then try and isolate and then try and prove that actually it's important for the bacteria to be there to produce the compound. So you do that. And then once you've done that, you do further kind of colonization of germ-free mice, you do further metabolomics, and then eventually you do some gene knockout models where you knock out specific genes which are critical in producing the compound. And then eventually you might be able to prove that microbiota is indeed what causes the disease, and then come up with microbiota-based therapeutics. So as you can see, that's an incredibly complicated thing to do, and there are a number of different variables which could go wrong. And despite trying, we've not been able to do it in the field of inflammatory bowel disease, for example, where we've been able to narrow it down to a single pathogen or a set of pathogens. And the best we've got to is things like E. coli and Fecal bacterium, where it predicts disease recurrence or disease with modest sensitivity and specificity. And when you try and get rid of these organisms with antibiotics, it doesn't necessarily work. So we don't have a fully functioning, causative mechanism for gut microbiome for huge populations of bacteria. It works very well for single bacteria, but not for bacterial community disturbances. Next, we go on to therapeutic modulation, and I think this is quite important. If you can't do the Cox postulates, then the ultimate sort of proof that you can get is, of course, a randomized controlled trial, where you do a randomized trial and then you get rid of certain bacteria, or you do whatever antibiotics or etc., and then show that that treatment works. And that, of course, is the ultimate proof, and of course the one that's completely clinically relevant to practicing physicians. So how can we achieve, how can we modulate the gut bacteria? And of course, there is a number of different ways you can do it. You can do it in quite an untargeted manner, meaning that you can just generally promote interventions that might promote a healthy bacteria, for example, exercise, prebiotics, probiotics, and of course, fecal bacterial transplantation in the hope of changing the entire bacterial landscape. And within that, there are things like prebiotics. Sorry, I've got these two mixed up. So probiotics, where you can get specific organisms. These are live organisms, and you take, and people can take it. And this is a multi-billion-dollar industry across the world, where there is a lot of health belief in probiotics, but the science necessarily supporting probiotics is not fully there yet. You can also modulate bacteria with prebiotics, things like inulin, for example, or non-digestible fibers, with the hope that fiber-generating bacteria like Prevotella may produce short-chain fatty acids and may be of general health. You can also do it with a combination approach called symbiotics, where you mix prebiotics and probiotics. Or you can also use this thing called postbiotics, which has been explored in many different fields, including, for example, C. diff, where you can take individual bacterial strains which are pasteurized or killed, or get the bacterial products as supernatants. So without the bacteria, you just get the products that the bacteria produces, and then use that to change the milieu of an individual. You can also do it in much more sophisticated manners. For example, you can do specific targeting, and I think in IBD, this has been done by using a probiotic bacteria called Lactococcus lactis, and that is genetically engineered so that it produces interleukin-10, which is an anti-inflammatory cytokine. And this has been done for diabetes, injecting the glucagon-like peptide 1 for diabetes, where you engineer probiotic bacteria to produce compounds of interest. You can also do more futuristic things like phage therapy, which is bacteriophages to eat away certain microbiota which are bad, specifically, and also maybe in the future gene editing, which is already being done for certain diseases. So let's look through some of these things. Diet, among all those interventions, diet is probably one of the easier ones, not necessarily easy to follow, but at least easy to formulate. And there have been lots of different diets which have been shown to have an effect on bacterial population, as well as an effect on symptom relief. For example, if you take irritable bowel syndrome, you can put people on low FODMAP diet, fructans and oligodisaccharides and polyphenols. If you reduce the amount of that, it increases certain bacterial populations like actinobacteria, and also decreases the abundance of certain bacteria which produce gas. And it also improves the symptoms of irritable bowel syndrome. And there have been a number of interventional studies, and more recent studies have shown that actually, if you have a certain bacterial population within your gut, you're much more likely to respond to FODMAP diet as compared to a different set of bacteria. So there is already precision medicine happening using diet as a way of modulating bacteria. This is, of course, done with a number of other things, vegetarian diet, Mediterranean diet, etc., etc. But suffice it to say that actually diet can be used as a way of successfully altering bacterial population and improving symptoms. Probiotics, I've already mentioned probiotics. These are live organisms which can actually, not only can they change the bacterial population, but more importantly, even if they don't change the bacterial population, they can change the dynamics within the community structure. They can also produce certain compounds which themselves might be of benefit. And probiotics have been shown to be helpful in a number of diverse health conditions. But predominantly, like for example, C. diff, and necrotizing enterocolitis, for example, there's some evidence to suggest that probiotics may work. Now, the difficulty with probiotics in general is the fact that there are a number of different probiotic bacteria. For example, Lactococcus I've mentioned previously. Then there is things like, for example, Faecalibacterium or Saccharomyces boulardii, lots of different things. And because there's lots of different things and there's lots of different dosing regimes, it's very hard to be conclusively certain whether or not probiotics produce meaningful benefits for chronic diseases. Say, for example, in inflammatory bowel diseases, for example, there have been a number of probiotic studies which have not really shown any benefit apart from maybe a small study using E. coli Nissel. But otherwise, it's not been, probiotics haven't been shown to be a particular benefit in chronic diseases. But in certain conditions, for example, C. diff, I think there is moderate quality evidence. Then we go on to fecal transplantation, which I think if you, we've spoken a lot about what constitutes healthy microbiome. We have talked a lot about how the microbiota has changed in disease states. And of course, we have talked about the fact that you can transfer the phenotype, at least in mice, by taking microbiota from obese mice and putting it into lean mice, and vice versa. So that lends itself nicely to the hypothesis that actually, if you can change the bacterial population fundamentally by using fecal transplantation, then you might be able to actually influence disease severity and disease course. And of course, this concept is not new. It's been practiced as early as the 4th century, apparently using oral human fecal suspension called yellow soup. Sounds dreadful and reminds me a bit of dal makhani, but nonetheless, people have used it in China, and I can't imagine how it would be to drink somebody else's feces to cure an illness. But thankfully, we've moved a bit further on, and the technique has been refined, and people no longer have to eat or drink it, but you can give it as an enema, or you can give it through an NJ tube, and so on and so forth. And the landscape completely changed with this randomized trial that was published in New England Journal some years ago for C. diff. C. diff, of course, is an illness that's caused by overgrowth of a particular bacteria. And it happens typically in older people who are frail and who have had antibiotics, and where they have a dysbiotic population. And older people who are undernourished tend to get recurrences of C. diff. So in this study, what they did was to randomize patients to either fecal transplant, which is administered through an NJ tube, nasojejunal tube, or just continuing with vancomycin with the primary endpoint of Crohn's disease relapse. Clostridium difficile relapse. And the data monitoring committee for this particular study had to come in and then say, let's stop the study early because the benefit was overwhelming for fecal transplant. So this was the first disease where fecal transplant was officially shown to be beneficial. And of course, now it's a licensed treatment for C. diff. And this has been proven time and again in further systematic reviews. This recent one was published in Lancet a couple of years ago, again showing that now we are looking at the finer points of fecal transplant, whether you should give it as a single dose, whether you should give it as a repeat dose, should you give it as a capsule, should you give it as an enema, should you give it as a NJ tube, or should you administer it through colonoscopy, and so on and so forth. And there are some subtle differences depending on what you do. For example, if you give it through the colonoscopy, you administer the stool, that is much more, probably a little bit more efficient than giving it through an enema or an NJ tube, for example. But that's for a single illness caused by a single pathogen. And now we can look at what we know about fecal transplant for chronic diseases. So I'm going to stick predominantly to inflammatory bowel disease. So there have been a few studies that have been done now, a few randomized controlled trials, and this is the first one, which is published from Moyadi and colleagues from Canada, where they randomized patients to either placebo or weekly for 6 weeks. And importantly, these are from single donors. So donor A, donor B, or donor C. I think there were 3 donors in the study, and there was a difference in the primary endpoint. So more patients in the fecal transplant group achieved remission as compared to people in the placebo group. And interestingly, there were 6 donors, but most of the improvement was noticed from one particular donor called donor B. And subsequently, they published some studies to show that donor B had a slightly different set of bacteria compared to the other 5 donors. But the point that this underlines is that actually donor selection is probably a key attribute for fecal transplant to be used successfully. And then there was a further large Australian multi-center study. The good thing about the study was that they saw the findings from the Canadian study, and then they modified their protocol such that actually you're not relying on a single donor contributing to the success of your study. Instead, what they did was they got stool from 3 to 7 donors, and then they mixed it up in a big giant jug, I would imagine, and then they went full on in terms of treatment. So for the first time, they put in the fecal installation through a colonoscopy, and then all the patients had enemas 5 times a week for 8 weeks. And again, they showed a difference in terms of the response rates remission rates between fecal transplant and placebo. So there was around a 19-point difference, which is actually very good and very similar to what we see with some of the other biological drugs for inflammatory bowel disease. And they subsequently did some bacterial population analysis. Here's the operational taxonomic units and the diversity that I was talking about before. So if you focus maybe on the diversity graph, you can see that at baseline, patients who had a diversity which was lower, but once they get the fecal transplant at week 4 and 8, and at final follow-up, the diversity increases from a baseline of here to here, i.e. much more bacterial species. Compare that to the people who got placebo, there was no difference in terms of diversity at week 4 and week 8, which is what you would expect on baseline. More or less things stayed the same. And then the diversity of these people who had the fecal transplant was very similar to individual donors, but not as good as the donor batches. At least they were able to achieve some of the therapeutic effect. But of course, again, we get into this chicken and egg situation where whether or not the improvements in diversity are associated with disease remission as compared to just the effect of fecal transplantation. And they were able to show some independent effects of improvement in diversity outwith the remission. It's also been shown to be successful for other conditions, like, for example, autism spectrum disorder, which is an open label study. So if you take it with a bit of skepticism, obesity, I've already mentioned. There's some preliminary exciting data from primary sclerosing cholangitis. Irritable bowel syndrome, the evidence has been mixed, one study showing good effect and one showing bad effect. But there are some studies in, for example, hepatic encephalopathy, and there is an ongoing randomized trial of fecal transplant versus placebo in hepatic encephalopathy, where the main problem is production of metabolites such as ammonia, which impair brain function. But there are several different unanswered questions about fecal transplant. Firstly, we don't know which route to give it in for chronic diseases. We don't know how often to give it. Do you have to give it lifelong? Do you have to, like for example, in the case of inflammatory bowel disease, you need to take long-term medications. Do we think fecal transplant is exactly the same? But more importantly, I think this question has not been answered yet as to what are the donor attributes. So unlike sperm donation, where there is an obvious couple of attributes, for example, the donor has to be good-looking, intelligent, and handsome, and then you'd have a bit of the sperm. But for fecal transplant, you can't look at someone and then say they've got good diversity or bad diversity. So we don't know whether the donor is an important part of it. And there are some ongoing studies which might address some of these aspects. Also, we don't know whether you need live bacteria or bacterial products. And of course, we have to remember the fecal transplant is licensed, for example, in the UK as a medicine, and therefore we need to be prepared to experience adverse events. And there were the highly publicized cases of E. coli, ESBL, septicemia after fecal transplant. And after that, the US FDA put a halt to kind of DIY, do-it-yourself fecal transplants. And these days, it's a highly regulated industry where you have to pass through some checks to make sure there's no transmissible pathogens with fecal transplantation. So I think with that, I'm hoping we'll come to the end of it. So just to conclude then, gut microbiome is an increasing area of focus for many, many different research groups. I've not gone into it as exhaustive a detail, partly cognizant of the fact that there are clinicians from multiple specialties. Diverse associations have been shown between gut bacterial changes and disease states. This includes dementia, Parkinson's disease, and of course with GI diseases, it makes intuitive sense, but with neurological diseases, you wonder what the causative mechanisms are. But that's why causality is still unclear. So we don't know whether the associations that we are seeing is simply an epiphenomenon or whether it's central to causation, and only time will tell. And randomized experiments of modulating the microbiota might give us a clearer answer. Therapeutic modulation by a fecal transplant has shown some preliminary evidence of success for chronic immune-mediated diseases, but like I alluded to, there are many, many different unknowns, and the field is ripe for further investigation. And I think with that, I'll conclude and happy to take any questions. I'll answer the simple questions and leave Bala and Dr. Ramakrishna maybe to answer the more complex ones.
Thank you, Sridhar. If you can stop sharing your screen.
Yeah, great, will do.
And your comments first, Dr. Ramakrishna.
That was an excellent talk, Dr. Sridhar. I enjoyed that. It was it kind of going through the gamut of the gut microbiome and the variety of things it does. So I think there's actually been, as you said, I think there's been interest in this for many years. I mean, the technology has advanced in recent years. In fact, I think about 25, 30 years ago, with the soil microbiologists coming into their own, with the molecular microbiology, that's when we started this. In fact, Bala and I did some of the original studies in India which used these kind of molecular microbial methods.
Yes.
There's a big gap between what we hope we will get out of the microbiome and our current understanding of how to manipulate. Isn't that right? But so we know that probably a lot of neurological diseases may have something to do with gut microbiota. We know that aging may have something to do with the gut microbiota. We know that nutrition has something to do with the gut microbiota. But what are we going to do about it, or how can we get? I mean, I think you've highlighted the technical difficulties there, and how to fulfill Koch's postulates in a modern
Yes.
molecular biological kind of way.
Mm-hmm.
So I think I'll just ask Bala to comment as well, and then I'm sure there are questions from the audience that we might get to.
Thank you, sir. Firstly, thanks, Dr. Vidya, for inviting me. In fact, you should have. I would have been happier if you'd have introduced me as Dr. Rama's student than the list that you mentioned. Thanks, Dr. Sridhar. I mean, it's a lucid and easy to understand for all talk. I think me and Sir, when we started working on this, I still remember the first day when Sir told, "Hey, there is this bacteria in stool, and nobody is interested in it." And so this is in 2002, and I told Sir, "Why not? I'll take this up." And that's how and I still remember all my seniors make fun of me, saying that he was sitting on a gold mine. That's the stool. So that's how the joke goes during my time. So I worked with Sir on various aspects of characterizing microbiome in Indian population. But slowly, my interest now is more of studying under nutrition biology, looking at or designing various interventional strategies to manipulate microbiome in this children who are undernourished. And I also work with Sir on various aspects like we do a lot of simple feeding study in terms of
Sure.
Prebiotic feeding studies and so on for many years now. So we have a lot of promising results.
Yes.
Starting from children to HIV patients who have managed to have widely suppressed HIV patients by just doing some simple feeding of prebiotic.
Sure.
So there's a lot of potential, and then we have in fact there are a lot of plans to work on various microbiome-based intervention strategies.
Sure.
And there are some slides that makes me very nostalgic, especially I'm sure, Sir, you remember that the Orlet slide that he showed, Dr. Orlet from we met her in Clermont-Ferrand, and we had a discussion on how she explained us how this E. coli sits in the
Arlette
Duffy,
yeah, who sadly passed away, didn't she?
Unfortunately, she passed away.
Yeah, that's right. Yeah.
Yes. So thanks again for this presentation.
No problem.
I think the topic is a bit too complex for a layperson to be just asking questions. So maybe either of you could just, emphasize on some of the things. And we have Dr. Reba. Today's program is thanks to her, because she suggested we should do this topic. And that's when I went looking for people who, would be able to tell us about this. Dr. Reba, did the session match your expectations?
Yes, it was a wonderful talk by Dr. Subramanian, and he really unraveled a lot of mysteries associated with microbiome. But as a clinical microbiologist, I have just...
Ma'am, you've got muted. You'll need to unmute yourself again.
Hello, can you hear me?
Yes, we can.
Yeah. Sorry, the question, my worry actually is, being a clinical microbiologist, is the transfer of drug resistance in the fecal transplant, which he did mention. Dr. Subramanian did mention about, transferring drug resistant genes through the fecal transplant. So how do you look at, the problem of this? And how do you think that can be contained or controlled?
I mean, that's a very good question, actually. And in fact, some regulatory agencies have taken a very stern stance about this. In the UK, for example, the Medicines Regulatory Agency have deemed this as a medicine, which means that there are only 1 or 2 accredited centers which can actually act as fecal transplants. I remember in my early years, we used to do fecal transplant from living relatives, healthy relatives. And actually that's, come to think of it, it's a bit of a crazy practice really, because we had screened for maybe a couple of things like HIV and hepatitis, but there's a whole bunch of other things that we didn't think about. But now there are some robust mechanisms for screening for known pathogens, known transmissible organisms. But of course that only, we can only screen for what we know. And there are some unknowns which we don't even know about. Given the complexity of microbiota, I think it is a bit of a dangerous thing to be sort of saying that I'm going to transplant 1,500 different bacterial species with virtually unknown functions. But I think we need long-term sort of safety studies to be able to tell us whether or not what we're doing is right or wrong. And for now, I think we follow best practice and then follow guidance that have been set out by different organizations on what we should do, and make fecal transplant a medical product rather than a do-it-yourself. There are some groups of population, particularly vulnerable population, for example, people with autism spectrum disorder, where parents are looking for frantic answers and they do things off their own back. And so you're completely right in saying there are particular concerns and we don't know. I think the field is too nascent to be completely confident about long-term outcomes. But I think we can say that short-term outcomes, we have not seen any adverse outcomes. We've done quite a lot for C. diff. But of course, we haven't followed up these people as rigorously as probably we should. And we need long-term safety data for sure. Whether there are transferable phenotypes, whether it's good or bad, whether it makes you an angry person if the donor is an angry man. So we don't know those kind of things yet. Only time will tell. I think that's a very valid point.
And one more question, which I think one needs to look at, is the presence of non-cultivable microbiota. The non-cultivable, what you can cultivate, you can look at, and you can look at the genes and then, characterize. But what about the huge non-cultivable?
Yeah, absolutely. That's a completely valid point. In fact, the field is advanced. Yeah, largely due to, largely due to sequencing technologies like we have pointed out. So the non-cultivable bacterial is a huge challenge. And in fact, even Faecalibacterium prosnitziae, which is widely touted as a probiotic bacteria, it's an anaerobic bacteria with stringent requirements. So we don't know how to deal with those things. Even if we come across what we think is a good bacteria, we still have the next challenge in terms of cultivating it, harnessing its benefit, etc. So you're completely right. We had the same problem for Helicobacter for a long time. So again, I think time is going to be key. I think wait and watch.
Yeah, there's a lot of work to do out there, and it's a very challenging field. Thank you so much. It was a wonderful talk, and thanks, Vidya, for getting them on board and sharing their vast knowledge. And thank you, Dr. Subramanian.
Oh, yeah, no problem.
So there was a question on the chat box from Sree. I think many of the aspects that probably would come under ethical concerns were addressed. Sree, if you have any specific point that you want to make, please unmute yourself and speak. Sree, you had a question on ethical concerns of. Okay. So looks like she's not ready. So Ravi, you have your hand raised.
Yes, thanks, Vidya. So the question that I had is that there are a lot of, when you look at all these large clinical trials, there's a wide difference in responses. So if you take GLP-1 receptor analog trials for type 2 diabetes, you have a huge variability in terms of how A1C responses occur, how weight loss occurs, etc. And I do believe that at least with the A1C response, there is some indication that that the microbiome or microbiota may play a role in determining a response. So, I mean, how well has this been characterized in terms of responses to therapy? Because, I mean, when we look at average responses, they look fabulous, but then in any situation, you know, there's a big difference. And of course, the same thing goes for placebo. There are some people, you know, for whom you could sell the placebo and have fabulous responses, right? So how, I mean, has anybody looked at it formally, and, you know, what are the things?
I mean, that's a very interesting question, actually. In fact, you know, for example, I can quote 2 examples for this. One is the example from inflammatory bowel disease. I couldn't speak to the GLP-1 situation, but for inflammatory bowel disease, for example, a biologic drug called vedolizumab, and there have been some response signatures in terms of gut microbiota, which are associated with response. So if you have this particular bacterial signature, you're much more likely to respond. But the receiver operating threshold curves are fairly modest, the ROC curves, you know, the sensitivity and specificity. You can't pin your hat on it and say that this person with this bacterial signature is going to respond to it. And I highlighted some of that in my talk as well, which the immunotherapy related responses and adverse events. Again, firmicutes, for example, was associated with response as well as predicting adverse outcomes. So I think, again, the field is quite new, and we probably need more dedicated research to answer these questions. And again, we need to prove that missing link between associations and causation, whether or not you know, is it association just simply by guilt, or is it causative in terms of somehow modulating response to those drugs? I think we don't know, we don't have all the answers yet, but I think there are some examples from other fields where microbiota predict response and predict adverse events.
Right. I you know, because one of the big reasons is that a lot of people pin the variability in responses to polymorphisms in certain genes. And, you know, while they explain a certain portion of the variability, they don't explain a significant amount either. And so at this point in time, it doesn't seem to be appropriate to segment the population based on polymorphisms, because it doesn't completely predict or rule out responses, right? So so I'm just wondering if it's a polymorphism microbiome interaction.
Yeah, absolutely. I mean, you're completely you completely hit the nail on the head. In fact, there is this new ghastly emerging field of multiomics, which is essentially a hypothesis-free science where you take samples from every person, starting every treatment, and then put them through a sequencer and see what comes out, and then come up with a predictive algorithm. So for example, in inflammatory bowel disease, again, there's this huge European-funded multi-omic studies that are going on, which hopefully will explain some of the variability in terms of responses. And just in terms of what they're looking at single-cell transcriptomics at the mucosal level, in the blood level, in the microbiota, and so on and so forth. So there's a lot of huge consortia efforts that are underway to try and explain the variability by using multiomics, essentially.
Yeah, they cost a huge amount of money, but thank you so much for a wonderful talk, and you know, the additional information that the moderators brought into play. So thank you so much, and thank you, Vidya. Very fascinating.
Dr. Ramakrishna or Balamurugan, would you like to add anything?
No, I think we've touched on many of these things. One of the things that probably some of the earlier microbiota directed interventions, which is there in Ayurveda, it's there in other things, is basically colon cleansing. And this used to be, you know, very widely practiced, not only in India, in the US. In fact, I have a slide where I have a picture of a colon hydrotherapy center just behind our hospital in Madapayani, where they say it's done by expert doctors using imported equipment. And I just wonder what your take on something like that would be in terms of a microbiome-directed intervention.
Yeah, I mean, I think, yeah, I am not aware of any specific evidence of colon cleansing and health outcomes. I know that patients actually quite like it as a concept. They feel lighter, and there are some people who go for it, but I don't specifically know that there are robust health outcomes. It's not been tested in a randomized trial, as far as I know, unless you know.
So it's right for a randomized trial, you're saying?
It's not been, it's not been tested in a randomized trial.
Yeah.
I don't know whether we'll get any funder to fund it, hopefully. Yeah.
So, Bala, would you like to add something?
So my simple take would be that to look for the microbes, in fermented food, and the metabolites from these microbes in our day-to-day food that we consume. Because he mentioned cheese as one of those food diet to focus on, because cheese has about 9 to 15% of short-chain fatty acids that essentially what we want to increase by providing this prebiotics or other fibers that we use to intervene in all these conditions. So I think look for all these foods that are there on your plate, and focus on more of this fermented food, and look for foods that are high on short-chain fatty acids, like your butter. Like if you think that those are going to increase your lipid profile, disturb your lipid profile, then think of fermented rice water that we have very good results in terms of that has the lower concentration of short-chain fatty acids, but I think it has the right amount of short-chain fatty acids. So I think look around for these good microbes and their metabolites around us, and that's my addition to that. Thanks.
So Ravi had a question in just, does the stinkier cheese is the more stinkier the cheese, is it better?
I don't know whether.
Yes. So the stinkier it is, the more likely the concentration of butyrate is higher, and the bacteria which are there are actually those which ferment to give a lot of butyrate. And I understand that it's butyrate which attracts the mice. So it's particularly the stinkier cheese which attracts mice. But that's supposed. So there is a reason for believing that stinkier cheeses are better.
I don't think Ravi quite expected that answer. So Radhakrishna, your take on today's session.
As rightly said, it's an amazing talk said by Ravi Shankar. But then, this is too much for a surgeon like me. I would like to ask Dr. BSR a simple question, that this gut is such a dynamic organ that is all garbage in, garbage out. Where do these bacteria reside? Don't they is a flowing stream? Don't they get flushed out day in, day out? That's question number one. Where do exactly they reside? Everything that goes in should come out. And where do they hide, rather? And second thing is, on a daily basis, we remove parts of gut and throw it out. What will happen to this gut bacteria? Suppose you remove the colon, a part of small bowel, part of stomach. Will there be an alteration in the constituents of the bacteria?
Yeah, those are interesting questions. So the answer to the first question is that most of the gut bacteria actually live in the lumen. There are mucosa-associated microbiota, which are fairly adherent to the mucosa. There's the mucus layer on top of that, where a lot of bacteria reside. But a significant number actually live within the lumen. You have to realize that in Indians, the total gut transit time is 24 hours. In Western people, the total gut transit time is something like 48 or even 72 hours. So the colon is filled with a lot of fecal matter. We see these CTs every day, you and I, where the colon is filled with fecal matter. That's where the bacteria reside.
And
then
taking parts of
this,
that, will it alter the constituents?
So that again is an interesting. So that's where. So there was a British surgeon in the mid 20th century, Sir William Arbuthnot, and basically he was knighted because he did the large number of colectomies to improve people's health. So basically, he thought that you remove the colon, you remove the bacteria, you improve their quality of life. So, as Sree said, they didn't do a randomized controlled trial at that time. So.
Yeah, but also to say that you're quite right actually. So, for example, when you do an ileoanal pouch, the ileal bacteria soon acquire a colonic kind of phenotype. So there is some compensation for removed aspects of the gut, where the small bowel takes on a colonic kind of microenvironment, not fully, but partly at least.
So I should tell the BSR that in the hospital where I worked earlier, there is this gut flushing equipment. And there's a transparent tubing through which you can see water flows with pieces of feces, and the water is completely clean. The gut wash is done.
I'm sure that gives a glow to the brain as well.
Yeah. And maybe all the bacteria will disappear. Wonderful talk indeed. Thank you.
Anything? Any closing comments, Sree?
No, that's it. Thank you very much for the invitation, actually. It was great. I learned a lot as well in the course of preparing for it. And thanks for the invitation. Great job in terms of organizing it. I think it's a wonderful initiative.
Thank you, Sree, Dr. Ramakrishna and Bala for sparing the time. And thank you everyone for staying awake and actually taking in so much information, which most of it is new to us. We'll meet again next week with another episode of Marvelous Medicine. Till then, take care and stay safe. Good night.
Great. Good night.
Thank you.
Thank you.
Thank you.
Bye. Bye.